Now our next topic is describe the properties of monomers. Monomers are what? Individual.
Monomers are a group of like micromolecules.
Correct. That is the individual one. And the type of bonds that connect the monomers in biological to make macromolecules. That is
Molecules.
multiple.
Yes. Hmm.
Okay. That we have to study. That what type of bond is that? We have to study now bond. Previously, we have already studied about that components of our this one composition of our macromolecules, that is our what? Carbohydrates, lipids, proteins, as well as nucleic acid.
Hmm.
We have already studied it, right?
Yes.
Fine. Here did I written it what? That nucleic acid is a monomer of nucleotides. No, I have to mention it. Nucleotides are the monomers of nucleic acid. Okay, I will add it.
Monomers of nucleic acids.
Yeah, nucleotides are the monomers of the nucleic acid is what? Macromolecules, polymer,
Yes.
like that. And monomer is what? Nucleotides.
Nucleotides.
Okay. Here, one point is missing there. I will add it up. Now, there are three types of bonds are there. Peptides for protein, glycosidic bond for carbohydrate, and lipid bond for lipids. Okay, so one by one we will study. Now, peptide bond. Peptide bond is for what? Protein,
Oh, yes.
right? And what is
Yes.
protein? Protein is a combination of what? Is
Monomers.
formed by amino acids, right? Protein is a
Yes.
combination of different amino acids. Those are what? Glutamine, glycine, isoleucine, arginine, and betalanine. Right? These are
Yes.
our amino acids. So in our protein, we are having a bond which is known as what? Peptide bond.
That is a bond for proteins.
That is the bond for proteins. This type of bond is exclusively for the amino acids. Amino acid is what? Monomer, right?
Monomers. Yes.
Right?
Micro.
And our protein is what? Polymer. So these monomers create bonds between the carboxylic acids of the first and the amino group of second. You can see here.
So amino acids bond with carboxylic acid
Correct.
to.
That is COOH is the what? Our carboxylic acid. C double bond O, single bond O, H. This is our carboxylic group. Clear? It makes a bond with our what? Amino acid. Right? You can see here, it makes a bond with our amino acid. Now this carboxylic group will make bond with another amino acid. The chain will be like that. Clear? That's why it is written that the bond creates between carboxylic group of first, that is your first year, and the amino acid group of the second.
Auntie, what does of first mean and of second mean?
Suppose if I am saying this is Ananya, Ananya is first, Rashi is second, now it makes a bond, joining bond. So first or second is nothing. This is one amino acid, this is another amino acid. So one amino acid, take it as a first, in which this makes a carboxylic makes a bond with the another amino acid. Now, if there will be another chain, if there will be another chain, N, H, H, C, H, R2, R whatever R. Okay, then C, O, H, double bond O. Okay, now suppose if this one will be act as 1 and then this one will be act as a 2. Now this carboxylic group will make a bond with our another amino acid.
Oh,
Then this one will act as a first and another one will act as a second. Then this
second.
amino acid will make a bond with another amino acid.
Huh.
Clear? So
Yes.
C double bond O, OH is our carboxylic group. So here this part is clear.
Yes.
So first amino acid combines with another amino acid, then another amino acid act as a first amino acid, then combines with another amino acid. The chain will repeat it like this. So the connecting part, the connecting bond, this bond is known as what? Peptide bond.
Peptide bond.
Clear? So we have noticed some amino acid monomers have groups acid or amino into the primary ramification. It groups can't react to form peptide bonds. Very few, just like here. Very few, it can't be taken as a, can't be able to make a bond. Clear, bacha? Otherwise, most of it, majority of it makes a peptide bond. But there is some exceptions also. Why? Because of, yeah, say.
Can you repeat that one more time?
Sure. Very few, majority makes peptide bonds.
Oh, very few amino acids.
Can't able to make peptide bonds.
Huh.
Okay? That side is written, we have noticed some amino acid monomer. That is maybe in any one of them. Any one of them can't able to make what? Peptide bond. Have group acids because they are having group acids or amino into the primary ramification, and it is a primary one. So it can't make a peptide bonds. So it doesn't react with the another group and can't make the peptide bonds. Okay. And this is bonds, not bonds. Fine?
Yes.
Okay. Any doubts here in peptide bond?
No.
Very good. Now, the next is what? Glycosidic bond.
Carbs.
Now, glycosidic is for what? Carbohydrate. And our carbohydrates are what? In which we are having hydroxyl group. That is our monosaccharide. Monosaccharide is what? Our monomer.
Monomers. Yes.
Correct? One bond. One. So here we have already studied that in ribose, that is in our RNA, we are having what? OH, whereas in deoxyribose, that is our DNA, we are having H and OH that we have already studied. So in the same way, here is also making a bond. So this is what our DNA. H, OH, OH, H. Okay. Now, this bond is formed. So this is our first chain. This is our second chain. Clear? Now,
yes.
This bond is formed between carbon alpha of one monomer. It is this one. This is your what? Carbon. These are the carbons. Remember this thing. The carbon of one monomer, that is one monosaccharide, and the one hydroxyl group. Hydroxyl group we have already studied that it is what our C double bond O single OH and R. R is our carbon chain, right? We have already studied this thing. So hydroxyl group of what? C double bond O OH, and this is our R bond. So from this OH, it makes the group.
So
That is, one monomer combines with this OH. Previously, it is combined with what?
huh.
In our peptides, we are having this OH combines, this OH combines with H. Okay,
So there's two chains, Auntie. There's two chains, and there's a form between like the carbon alpha
yes.
and
Carbon
the
of one and
so one
OH. Yeah,
so one monosaccharide and one OH.
okay, fine. One hydroxyl group of another monomer. The number of carbon that has the OH group will bring the name of the bond. How many times it makes the bond? According to that, what number it is, just like in our glucose. Glucose number starts from here. This is 1.
Yes.
Okay.
One.
Now fructose. Fructose is having, this is also what our 1. But here, for example, glucose 1,4-fructose. But this example says that this is glucose. This is not the image of this glucose 1,4-fructose. This is not the image of that. If it is, if you want it, then you have to make it this one as a what? Our 4. 4, 3, 2, 1. And this will remain as a 5. From where you are starting the number. Just like, wait, this thing you doesn't know, no? That's fine. Just like here, the number is what? 1. Here also glucose number is what? 1. So the name will be what? Glucose 1,1-fructose.
Why would it be 1,1-fructose?
Because here's the position of carbon to which it is bonding. It is what? 1.
Yes. And
fructose, in which, see, this is 1, and the fructose by which it is making a bond, that CH, that is also what? 1.
1.
So we have to write the position.
So
If we have to make glucose 1,4-fructose, then we have to make a chain like this. It should be 1 here, carbon, H,
4,
then O, this should be C, H2OH, and then it makes a bond like this. Then here it should be numbering, should be labeling, should be 4 here.
4.
Understood?
Yes. How to label? Okay.
Okay. Now suppose this will make a bond. This will also make a bond, no? This is also
Yes.
for CH2OH. This is the first. Take it as a first. And here some another, our DNA is there. Ribose is there. Deoxyribose is there. Okay. So take it as a glucose. 2. Then it will be what? Fructose.
Fructose 5.
Fructose 6, na? Beta 6 पे bond बनता है ना?
Sorry, 6.
Fructose 6, and suppose if it is 1 glucose.
1 glucose.
Understood?
Yes.
Very good. This is the way to name it. Fine. Now, also we have the direction of the bond before it first number alpha or beta. It depends on the geometry of our monomer. Totally depends upon the geometry of the monomer, how we are numbering it. The main thing is the numbering. Okay, how we are numbering it, according to that we will, and numbering should be mentioned. According to that we will do our name. Okay, any doubt with this slide?
No.
Very good. Now lipid bond. Lipid bond is for our lipids,
Lipid.
in which we are having what? Fats. Lipids are having fats and cholesterols.
Hmm.
Correct? We have already studied it. Lipids are having what? Fatty acids and cholesterols.
So, yes.
Correct?
For
No. In this lipid bond, it is a bond is a covalent and creates an amphithetic zone. That is why some lipids are not soluble in water.
Oh, so like the molecules of the lipid do not bond with the molecules in water?
Right. Some.
Hmm.
Okay. And what is covalent bond?
Covalent,
Which is
well, isn't there two types like polarity and non-polar?
no covalent bond. See here. Okay, I have not taught you these things.
Isn't it polar covalent and non-polar covalent, Auntie? Like the two types?
See here, Peter. Hydrogen, lithium, sodium can donate how many electrons? One. We had discussed already.
Yeah.
So because of that reason, the bond which is made by these things are known as what? Mono. This can donate how many? Three.
Two.
Sorry, two. So di. This can donate how many? Three. So tri.
Sure.
This can donate how many? Four. So
Four.
covalent.
Oh,
Or tetra. Or tetra. Tetra or covalent bond. Trivalent bond. This is divalent bond. This is monovalent. Monovalent bond. This can accept, but here the bond is what? 5. 5 electrons in the outermost shell. The valencies of this one has 5 electrons. Valency is what? How many electrons are there in the outermost shell? So it will be 5. So this is will be this. Okay, wait. This one will be 6. So hexavalent. This is pentavalent. 5 means penta. 6 means hexa. 7 means hepta.
8
And
means
8
octa?
octa.
Octa. So it's like the shapes?
Yes. How many electrons are there in the outermost shell? That is a valence electron. According to valence electrons, we are naming the bond.
Bonds. Okay.
Clear?
Yes.
In it are mono, di, tri, tetra, or covalent, penta, hexa, hepta, and octa.
Yes.
Clear? Now, come to the slide. So here we are talking about the carbon chain. Here we are talking about carbon chain. Because of that reason, they are saying that it is bond is what? Covalent bond. Okay? And create an amphithetic zone. That is why some lipids are not soluble in water.
Anti-covalent means there are like 4 electrons in the outer shell. Okay.
See, all the carbons are making how many bonds? 4 bonds. 1, 2, 3, 4. 1, 2, 3, 4. 1, 2, 3, 4. See,
Yeah.
C double bond O, single bond R, and here OH. It means carbon has shared how many electrons? 4 electrons.
4.
And whenever we are having 4 electrons, it means it is what? Covalent.
Covalent.
Clear?
Yes.
Any doubt here?
No.
Very good. Now, lipids are... Yes.
What's an amphithetic zone?
See, creates an amphithetic zone. Amphithetic means